Thank you, everybody. It's a real pleasure to be here, even at 7 a.m. I think this is my third 7 a.m. lecture in 2 months. I'm not sure what's going on. So today I'm going to talk about charting a course from a human genome to personalized medicine. Okay, so I was told I had to give objectives. So the objectives are to convey the really Well, I'll give you a quiz. To convey the really breathtaking progress made in genome science since the sequencing of the first human genome, to communicate the multidimensional challenge of deciphering tumor genomes and transcriptomes, and to make clear the incredible challenge but also promise of personalized medicine. And this is thrown around all the time as if it's fairly trivial, sequence a genome and tell people what to eat and how long they'll live and when to have their prostate removed, but it's much more than that. But we're lucky because not so long ago, in a cave not too far from here, there's caveman and cavewoman, and the question is, how can we prevent pregnancy? We don't even know what causes it. Well, now, and really it wasn't that long ago that this was where we were with cancer, but now we know it's a disease of the genome, and we're fortunate that back in 2001, the human genome was sequenced in a 15-year, $3 billion effort that involved thousands of the best and the brightest. Now, in 1 week, with roughly $20,000 and probably in total 20 of the best and brightest, we can sequence the genome, transcriptome, and the exome to relative completion. So this is really quite a remarkable advance. And I think that soon you won't want to forget your iPod because it will tell you with certainty that Bill Gates is not related to Steve Jobs. So the question I've been working on for really, I guess, the last 8 years, starting out at UCSF in the Department of Urology with a prostate spore there, was can genomics be used to predict a man's risk of prostate cancer progression and metastasis? So this was an early attempt at personalized medicine. Could we take a prostate that was removed by prostatectomy, generate a genomic profile rather than an expression profile, generate a genomic profile, and predict the risk of a postoperative recurrence? So that's not. So now we're lucky at the Vancouver Microarray facility and innovation center. We have every technology you can imagine for studying gene expression, copy number changes and methylation changes on the genome, and because of the really brilliance of Sean Anderson, who I can't believe got out of bed and is sitting back here, we have protein arrays that are actually get published. Protein arrays have historically been extremely difficult to make reproducible, and he gets amazing data out of them, so you should come by and look. And if any of you want to do arrays or any kind of genome analysis, just come on by and collaborate. So we started out actually using a printer that was designed and built by Dan Pinkel, printed our own array CGH slides and analyzed a cohort of 64 prostate tumors. These are all at intermediate to high risk of having a post-operative recurrence. There were 64 tumors, 32 were non-progressors, 32 were progressors, and what was remarkable about this cohort was a 16-year follow-up, so that we knew the phenotype of these. We knew which were the benign phenotype and which were the lethal phenotype. And most of the progressors went on to bone metastasis, and that was confirmed by bone scan. So what I'm showing you here is more or less how array CGH works. It's actually interesting. I'm teaching my 18-year-old daughter how to do this summer. Well, she's an arts major, but she gets it. So the way these arrays are made in the old days is that we selected clones from across the genome, 3,000 of them, printed them on glass slides with a robot, and then extracted DNA from a reference genome, and from, I guess red would be the reference. Anyway, one color, green we'll say is the tumor, red is a reference, and then we hybridized those to an array of 3,000 clones. So we had roughly a coverage of 1.5 megabases across the genome, and then we could plot copy number from along the genome. So regions that are normal copy number would be 0, and things above, like this, would be amplified, oncogenes deleted, like here would be a tumor suppressor. And then Martin spent lots of money buying these expensive toys for us at the Microarray facility, so that we don't have to do printing anymore. So now you have up to 1 million of these probes printed on glass, and then use these sophisticated imagers to have extraordinary resolution. This is original data from the 64 tumors we looked at. So what we're looking at now is not gene expression. This is genome copy number from one end of the genome to the other. So 1P to YQ. And we're not looking at absolute copy number, but rather frequency in the cohort. So, for example, this amplification event happens in roughly 50% of tumors, and this deletion event is happening in roughly 40% of genomes. So we asked a very simple question, and that was, can we look in this data and identify a molecular profile, amplifications and deletions, that we can use to predict the risk of a post-operative recurrence? And we did, in fact, find a single genomic loci, which, when it was increased in copy number, predicted recurrence independent of stage and grade. And I'm showing you a box plot of that data here. This was subsequently confirmed by fluorescent in situ hybridization on tissue microarrays. And we even took it down to the level of inactivating the gene using siRNA and showing that in both prostate cancer cell lines and breast cancer cell lines, proliferation dropped. Remarkably, this is the multiple endocrine neoplasia gene, which is known to be a tumor suppressor in other cancers. So here we know in breast and prostate that it's behaving as an oncogene, but in other cancers it behaves as a tumor suppressor. There's now evidence from other laboratories that this is involved in tumor progression. But single biomarkers don't make the grade in heterogeneous cancers, and so I was very frustrated having all this data, all these tumors, and only one biomarker. So I think this points out the real necessity of hypothesis-driven science. Often you hear in genomics you don't need a hypothesis. Just go do experiments, spend a lot of money, and figure it out later. I don't actually think that's a good approach. So I formed a hypothesis. I call it evolutionary logic, and it's shown here. If we imagine that over here we have the primary tumor, it's a genomically diverse primary population. And as it goes through the process of metastasizing or moving towards the lethal phenotype, it has to pass through an evolutionary bottleneck, which I hypothesize would homogenize the population, the genomic population of these tumors. So, for example, an amplification event that's present at 20% in the primary tumor might be present in 50% of the metastatic tumors. And other events, for example, this blue event would just vanish. It would be of no value and would be lost in the process of metastasis. And so many of these selected copy number changes should be present in the primary tumor and maybe prognostic and even predictive. So we tested this. Now I've got a cohort of metastatic tumors from Ken Pienta at the University of Michigan. Did array CGH and compared them to our primary data. So this is just to remind you that in the primary data, it looked like this. Now I've drawn in lines at 30% gain and 30% loss, just to show you that the majority of events we're seeing are happening at well below 30%. Now, when we add in the metastatic data, we see that there's been an apparent homogenization. so that very many events are happening well over 30%. So for example, this event on chromosome 8, which may be the MYC oncogene, is happening at roughly 80%, and this event, which is happening on 8P, is happening at also about 80%. And I know many of you study prostate cancer, so you all know that this 8P loss and 8Q gain is really a signature of prostate cancer and bad outcome. But there's many others here too. For example, in addition to CSMD1 deletion and c-MYC, there's PIK3CA, there's CCND1, there's P53, there's RB. So this all makes sense in terms of what the genes are. So it seemed like the hypothesis might be correct. And so we went back and we asked, okay, well, what are these genes? And is there any way to develop a genomic panel that might predict a post-operative outcome? So this is what we call GEMCAP, so the Genomic Evaluators of Metastatic Prostate Cancer. And this just shows the data when we looked at the individual loci, which when taken together seem to predict progression and metastasis. So if we look across the top, this is, I think, 12 amplified loci, and the turquoise are primary tumors that never had a post-operative recurrence. Here we have primary tumors that metastasized to the bone and ultimately killed the patient, and in white is an independent cohort of metastatic prostate tumors. And so you can see for all the amplifications that this panel holds quite nicely, and the same for the deletions. We see this phenomena where the increase in frequency from primary tumors that never had a recurrence to those that had a recurrence to metastasis. Some of them, in this study, we didn't see aberrant in any of the primary tumors that never went on to metastasis. Now you can ask what they are. So this is Lymph kinase. This is a gene well known from in vivo studies and in vitro studies to be linked to metastasis. We have P10 here and RB here, and others. So this is, and one of them we found here we found the platelet-derived growth factor as being a bad indicator, and a similar study using expression microarrays at Harvard with Phil Febo found platelet-derived growth factor receptors. So they found the receptor, we found the ligand. So that was kind of a nice symmetry to have to suggest that we might be on the right track. So we identified these 39 GEMCAP biomarkers that appear to be able to capable of patient stratification. But I wasn't believing any of this. It seemed a bit too easy. So we went out and got another cohort of 29 tumors from the UCSF tissue bank. And we ran array CGH on all these tumors. We were blinded as to their outcomes. And then developed a very, simple algorithm about, how to interpret the GEMCAP profile. That is, we said if 20% or more are aberrant, that's bad. And if fewer than 20% are aberrant, that's good. We sort of picked that out of the air because we didn't know how else to do it. And what we found, in fact, was that we were able to go through that small cohort and very accurately pick out the guys who might be able to safely select active surveillance versus those that were going to have fairly aggressive cancer and who might be candidates for an intervention. And then we published this in International Journal of Biomarkers, and then Joan Hilton, a brilliant biostatistician from Harvard, now at UCSF, reanalyzed the data and came up with the same answers. So that was kind of encouraging. We then went on and did yet another study. This was on a group of, I think, 58 or 54 higher risk tumors. Exactly the same thing, array CGH on all these tumors. We were blinded as to the phenotype of these tumors. So we do array CGH, we apply our simple algorithm, and we got a little more sophisticated in this paper. And we just asked, could we stratify them Turns out that the GemCap signatures were able to detect the cases with negative lymph nodes that recurred within 1 year of surgery more often than the Catan nomogram, so that we clearly detect additional benefit of the GemCap genomic signature in predicting disease progression. And Mike Catan himself was kind enough to write a commentary on this work, but he noted we need to do another larger study, of course, always bigger studies. However, the GemCap has now been incorporated into a Uroscan, which is an array CGH-based scan for prostate cancer. This is not validated yet, but this is data showing DNA extracted from a prostate tumor, from formalin-fixed, paraffin-embedded tumor, in fact, just showing that we can pick up 8P, P10 deletion here, MEN1 gain here, and deletion of temperus here. So the hope is that at some point these arrays will get full-scale validation, and it will be able to predict a man's risk of metastasis from the genome. But what we really want to do is we want to tell guys before they have surgery whether or not they're at risk of progression to metastasis in their lifetime. But this is really challenging. I mean, some of it is challenging for reasons that you understand better than me, and others are just purely laboratory-based nightmares. So, I mean, you understand the problem of multifocality and heterogeneity in anatomy, making targeting difficult, and other aspects of it difficult. But what bothers me, in particular, are the very low DNA yields and the requirement to do this out of formalin-fixed, paraffin-embedded specimens, which makes this extremely difficult. So we can think about intratumor heterogeneity, that is, you have multiple foci of cancer within a single organ. Now, we've addressed this in one publication and in another study that's not been published. And what we found was that the concordance between different foci of cancer in the same organ is quite high. So on average, we found it to be roughly 91, 92%. In another study, probably a better study, using very high resolution arrays and really good pathology, we found it to be about 98%. So that looks good, right? But of course, the devil is always in the details with evolution. And I guess that 2 to 5% is more than what separates the monkey from Mr. Darwin. So this is so we don't actually know if it's good news or bad news that the different foci are so similar and yet differ by that 2 to 5%. Now this is the other challenge. So this is again something you're all aware of. This is a biopsy. Yeah, honestly, I expected to see a core of something like it came out of an 18-gauge needle. I was quite saddened when I saw something that looked like a hair in wax. But nonetheless, we have to figure it out. So this is 2 millimeters. It's 5 micron thick H&E, and we had to cut out the tumor and do array CGH. This is very challenging. We can make it work, but it's not. That's never going to happen. You know, going from sort of a biopsy to array CGH, right? You don't get enough DNA. It's not reproducible enough. So we have to come up with better methods. So fortunately, Informatics has this developed, commercialized, and turned into a service, the molecular inversion probe technology, which was developed in Ron Davis's lab at Stanford. And I'm showing you results with this. This is a great technology because it works with small amounts of DNA and doesn't mind if that DNA comes out of paraffin. So this is all from one prostate. This is chromosome 21, and this shows deletion of the temperus gene, which leads to the temperus erg fusion. And that's beautiful data. This is all from roughly 20 nanograms of DNA. So you can get this out of a prostate. This is so this is the biopsy here, run on adjuvant. This is analysis of the same biopsy using MIP technology. And so again, we see deletion here, but in addition, down here, we have allele specificity. So we can see whether it's mom or dad's chromosome that carries the deletion. And knowing that helps us have confidence in making the call in noisy data. So we have copy number and we have allele copy number. Then up here, we can see another foci of cancer that, or no, this is the radical prostatectomy rather, where we also pick up the deletion. So biopsy, RP. And then down here, normal. So it's flatline. So this is all good. So we're about to do this on 50 tumors up here in collaboration with AFI. And I'm trying to bring this technology into the microarray facility so that we can offer this as a service, because it's tremendously powerful. Because it opens up all the archives and biopsies to analysis. Now this is another possibility, which is to use whole genome amplification. So you get a little bit of DNA and make a lot of DNA from a little bit of DNA. And what I'm showing you here is work from Mark Magbuana at UCSF, and this is for circulating tumor cells. So this is whole genome analysis here on UCSF back array, so back to relatively low resolution. And it shows that the AR is not amplified pre-treatment. And then we look pre-treatment in 20 circulating tumor cells, and we see amplification of the AR. So it is certainly possible to use whole genome amplification, combined with, in this case, flow sorting, to obtain very small numbers of pure cells, and then do array CGH and detect events linked to the lethal phenotype. This simply shows one of the Combimatrix molecular diagnostics arrays analyzed in our lab, showing amplification at the AR, and this is visualized using software we have in the lab to make visualization and integration very straightforward. So again, this is a different experiment, so it is certainly possible to work with very small numbers of cells. However, an inherent limitation of all array-based technologies, whether you're looking at gene expression or genomics, if it's an array, it's averaging. So you think about, the 14 billion year history of the universe. It's like just focusing on the last few billion years and saying, we understand it. So, this is always a problem. You're always going to be looking at the dominant clone that happened recently. Moreover, it's like trying to understand the universe in a single wavelength, right? So you got all this other stuff going on, and you're looking like this, and so you're never gonna get there. So using sequencing, we're implementing next generation translational oncology at the Vancouver Prostate Center with Martin and others, and so this shows a pipeline that Stas Volek and I have put together with the help of a lot of people. And it's a pipeline for next generation biomarker and target discovery. So the idea is that the surgeons and doctors, Martin Gleave and Kimchi, and hope others of you, bring us tumors that goes into the pathology core with Ladan Fazal, and then we analyze these tumors by arrays, QC them, make sure that we get genomic profiles that look good, and then we can either do biomarker analysis on those, but mostly we're feeding those into an Illumina sequencer and sequencing the tumor genomes, and then doing bioinformatic analysis to identify biomarkers that can go on additional arrays, because arrays are always going to be cheaper than sequencing, so we can discover biomarkers here, print them on arrays, and then translate that to the clinic. At least this is the hope. This is the way we do it. This is Illumina sequencing. This is called a flow cell. We'll skip all the details. What's important is that with 4 lanes on this flow cell, we can sequence the genome to 3-fold coverage. So it's like sequencing the human genome 3 times. We can sequence all the exons. So every single exon in the genome, 50-fold. And we can sequence the transcriptome, so all the expressed genes, 40-fold. It's still amazing. And doing this, we can look at copy number variants, amplifications, deletions, inversions, insertions, genome breakpoints, and translocations. With the exome, we can see sequence variants, mutations, and fusion genes. And with the transcriptome, a similar amount of information from splice variants to mutations to sequence variants to fusion transcripts to RNA editing. Now, this all leads to what I call the new central dogma, which is we're bottlenecked, right? So these instruments have tremendous throughput, but this diagram shows the problem. So, over here we've got to do computational analysis and validation. So we sequenced a tumor from one of Martin's patients, and the exome alone generated 1,000 sequence variants that are classified as highly interesting. We've also got, I guess, 50 to 100 potential fusion genes, and what do? So we need post-sequencing technologies that can match the throughput of the sequencing technologies. Just computational analysis, every time I turn around, we're out of computer space. The Genome Sciences Center has a petabyte system, a petabyte, right? So this is a real problem. It's just the new central dogma. A lot. Stas is sitting right behind me. He's the guy with the details. Yeah. And now they're working on zeptabytes because petabytes are too few. So again, hypothesis and outcome-driven science is critical for translational. Actually, I think for any kind of science, but particularly for translational science. So, we're really focused on hypotheses and outcomes. And we have this cohort of tumors, and right now we're focused, or I'm going to talk to you now about studies with matched primary and lymph node metastases, because we want to understand the lethal phenotype, what's going on as it moves from the primary tumor to different organ sites. And what I'm showing you here, this is patient data. So this is what you'll have on your iPhone at some point. This is matched primary and lymph node. So on the over here is the primary tumor, and over here is the lymph node metastasis. So, if I look at this, sort of a seasoned array CGH person, I would say this tumor is not too bad. If I look at the MET, I'm thinking not good. So the red arrows indicate 3 chromosomes, 6, 7, and 8, that in the primary look more or less okay. I mean, there's some changes. If everything is good, you'd expect it to look like this region here. Tight cluster around 0, that's good, normal copy number. Start seeing things like this, not so good. But you start seeing things like this in a lot of them, say chromosome 2. Look at this evolution from here to here. I mean, this is just about a few centimeters in space. And then chromosome 6, 7, and can you see 8? And then chromosome 8, which I'm not sure if you can see. I guess you can. That's really diagnostic of bad behavior, loss of 8P and gain of 8Q. This is never a good sign. Now, we need to develop ways to sort of smooth the data, get rid of artifact, and help us see the differences. And so Robert Bell in the group developed this simple mathematical method, log of the ratio. So here we have I guess this is the primary tumor here and the lymph node here, and he puts it through this little algorithm of his, and it really teases out the details. So again, this is chromosome 7, and we see gain of 7Q here, and then we see this narrow amplification here. And the resolution is so high. This is, I think, 100 million reads compared to like 3,000 backs on the original probes. So we were making 3,000 measurements before, now we're making roughly 100 million. So we know we can just point and click on the computer and know that this is the EZH2 gene. That's interesting because EZH2 downregulates ECadherin, which is a cell adhesion molecule, and might allow these cells to migrate. We do FISH with David Huntsman at CTAG, and sure enough, it's amplified. It's the control probe here. The red is EZH2, and so we can see amplification. So that's great. So copy number changes, what are they good for? We can identify prognostic and predictive biomarkers. We can identify susceptibility and resistance genes. We can identify therapeutic targets, and we can get at mechanisms of resistance. An example would be ERB2 and Herceptin. Now recently, Anna Lepuk in the group has been working with, again, whole transcriptomes. So we sequence every single gene expressed in the tumor at deep coverage, 40 to 50-fold coverage. And so we can ask a lot of questions. Simple questions, what genes are expressed at high level. More interesting is what genes change their pattern of exon utilization, so alternative splicing, as the tumor goes from the primary site to the metastatic site. So this is an example of the androgen receptor in a patient here, just showing that this patient expresses a specific isoform of the androgen receptor. And so these are the, I think, yes, the yellow is what we call the default isoform. So this is what would be normal. So you can see these high levels of expression at the exons. Right? So now we're looking along a gene, and we're not looking at copy number, we're looking at expression of individual exons. So here's an exon, here's an exon, here's an exon expressed at high level, and these are the normal isoform of the AR. But down here in red are other exons that are expressed in this patient, for example, here, or no, not there, here, that we think indicate he's expressing a different isoform. And this is important because evolution of the lethal phenotype and castrate resistance can involve alternative splicing of the AR. So this is early days of our analysis. This certainly shows it's possible. Here's another example of the fatty acid synthase gene, which it's a little more clear, at least for me, where this is the urethra. This is the urethra here, and this is a penile met here. This is a neuroendocrine tumor. So here you see expression of this exon in the urethra met, expression of this exon, and this exon here, and these are essentially absent in the penile met. So again, this is a switch in isoform of the protein going from one site to another. Here's another example. This is the basal cell adhesion molecule, where we can see that they share these are the gene exons down here. So 3' UTR and a group of exons here, pretty much concordant. Here we have concordance, but the minute we get to the 5' region of the gene, something changes. So this was found purely computational. Stas went into the data, dug it out, pulled it up yesterday. I did a search. It turns out there's 2 isoforms of this gene expressed. Yes?
Just the point, this is a guy who had multiple metastases from a neuroendocrine tumor took out. And so here, I go back, that's a penis and a urethral lesion, and yet they're so different.
Yeah.
And it's the same patient dying of this one cancer. So you're showing how the more you dive down into this, perhaps it's this heterogeneity problem.
Right.
What's the driver and what's the passenger?
That's right. No, we don't know here. This is brand new data. But the AR certainly points that we can identify them. So, what can we use splice variants for, right? We can identify tumor-specific prognostic and predictive biomarkers. We can identify tumor-specific therapeutic targets, and we can get at mechanisms of resistance. We know that from the AR. And examples are BCLX. So there's a cancer-specific long isoform of this, which is an anti-apoptotic form, and there's a short form, which is an apoptosis-promoting form that correlates with resistance to chemotherapy. So I won't bore you with the details of this slide, but Wu, postdoc in the lab, has been we're working with people from David Huntsman's lab and a lab up at Simon Fraser has been identifying fusion transcripts. These are what we're finding is extremely interesting. But the bottom line is that these fusion transcripts, or the bringing together of genes that don't normally get together, is important because it will allow us to identify tumor-specific prognostic and predictive biomarkers, identify tumor-specific therapeutic targets, and again, get at mechanisms of our resistance. And I think that the data he's getting that I'll present at another time is going to greatly expand our knowledge of the genome's complexity and plasticity. And of course, the best example of this is probably the Philadelphia chromosome, the BCR-ABL fusion, which generates the BCR-ABL fusion, which is the target for Gleevec. So this is clearly where we want to get to. The other thing that we can do, sort of at remarkable speed, I think it takes roughly a week to analyze the data, but we can identify all the splice sequence variants in a tumor. So this is just an example where using computational tools developed by Sohrab Shah here at UBC, was able to take our data and identify all the potential mutations and sequence variants, and then classify them. So the ones in red are, interesting. They're radical changes. They should make a big change in the protein. And some of these are linked to genes that might be interesting. So, we get, I think the patient we just did, we found 1,000 of these. So we need ways to validate them. Now we can classify these. We can say, oh look, 44% of these are related to cancer, 23% have stem cell characteristics, so on and so forth. So that can help a little bit. And using other tools developed by Robert Bell, we can ask, in the primary and the lymph node met, or in a population of primaries and a population of lymph node mets, what's different. And so we can find events that lie off the diagonal that are different. And because this is all sequence data, every one of these little black dots is sequence data that we can identify. We can ask what they are and manipulate this. So we can feed what's different into network analysis. And in this case, this is using Ingenuity and patient-derived networks. We see, I don't know if you can see this or not, but the central, all roads here lead to PI3 kinase in this tumor. I think this is the neuroendocrine tumor. So, we can collapse all these differences down into common pathways that all seem to be converging on PI3 kinase. Now, what I think is particularly interesting is that we can take all of this data and we can build a genome and network database that we are building at the Vancouver Prostate Center. And then we can interrogate a therapeutic database. This Cyclips database is one I've been looking at. It seems quite useful. So we can say, okay, we've got these genes or mutations or fusion transcripts or alternative splice genes that seem to be related to progression in some way. We can go into this database and very quickly identify drugs that have been developed against those molecules, get all the patent data, and figure out how it works. So I think this is a real opportunity and something we're pushing on. The few molecules I put in here yesterday, I think 3 out of 4 produced a drug that's been evaluated and there's a patent on. So this brings me to a vision of personal genomics to therapy, where the doctor interacts with the patient, gets some DNA or RNA, this is an ion torrent sequencing technology here, grabbed off the web. We sequence, we get at the correct pill or therapy, and I think that increasingly if these are going to be genome-based or DNA, RNA, something like that based, because everything we're finding is nucleic acid targets. And to show this is more than just a nice slide. Again, this is work going on at the Vancouver Prostate Center with YZ Wang. We're actually profiling patient tumors. At the same time, those tumors are implanted in the renal capsule of mice, and we're evaluating the response of the tumor to drugs that are predicted based on the genomic profiles. So far, we're not having great results, but we just started. So we can grow a patient's tumor on the renal capsule, profile it, make predictions, and test those predictions in the mouse. So I think this is very exciting. So with that, I'll end and take questions. Thank you.